Hello and welcome back. Beyond Networks, the evolution of living systems. I don't know about you. I've had a lot of excitement the last week and the weeks before. We went on a really broad exploration of how to study evolution. What are the tools that we have? What are the things we've already done? What are the things we can do? And so the last two modules of the course are going to explore the question. So what should we do next? Okay. And so we've been encountering these really big challenges, like, the challenge of open evolution and radical emergence. In the words of Stuart Kauffman, evolution is radical, ongoing co-creativity. The rules always change. There's context-dependent co-evolution of states and their constraints. They co-generate each other. And not only can't we predict specific future events, but often we can't even know what can happen, because the rules always change, because we have no probability distributions, therefore, based on which to predict future trends. And this is a really, really deep challenge, as I've tried to convince you, because we need to completely change the way we do science. And so Lee Smolin has formulated it like this. We have to go beyond Newton's paradigm. We're doing physics in a box. And this is how you do physics in a box. And Newton did this for the whole universe, basically. If you want to understand the future trajectory of a system, or if you want to trace it in the first place, you need to specify the initial conditions of the system, the boundary conditions in time and space, that's the box, the physics in a box approach, and the rules of change, like described by differential equations, for example, that apply within the box. And they won't change usually, or change in very predictable ways. So that, again, defined by rules. So in this sort of approach, there is no true emergence in the sense that there are no new rules or configurations that can come up that weren't predefined before, at least in theory. That's the problem of the symphony, of course, again. There can be very large configuration spaces. So we need to move beyond this, but this is going to be very, very difficult. And we're only beginning to even think about this. So in the very last module, not this one, the next one, I want to explore a little bit, at least the very sort of broad strokes about what could a science look like? What kind of explanations would it seek? What kind of questions would it ask if it would really go beyond Newton's paradigm? And we wouldn't recognize it necessarily as science like we do right now. So we have to explore this very carefully and philosophically before we can do actual post-Newtonian science, if you want. But for this week, we want to stay a little bit more on the ground in current reality, in the present. And so let's explore what we can do with the tools, these Newtonian tools that we have right now. And what is there left to do? And so to illustrate what is left to do, we can look back at these sort of four different perspectives we can take on evolution: structural, functional, process, agential perspectives about the influence of the goal-oriented behavior of the organism on evolution. And we'll realize that only the fourth one, the agential perspective here, requires this post-Newtonian approach. So what is left to be done here? I think structural and functional perspectives have been very well explored in evolution, but let's look at process perspectives. My own sort of past tradition is process structuralism. I was a student of Brian Goodwin's, and there's developmental systems theory, there's the reproducer perspective that James Grissomer has proposed, and these remain hugely underexplored. So let's have a look at what we can do. So these process perspectives are interested in how do processes evolve, right? So the unit of evolution is a process such as a life cycle or a developmental trajectory. And these are described by generative processes in the morphogenetic fields that sort of underlie them, okay? So what we need there is sort of an idea of how these processes are, what they are in the first place, and then how they can evolve. So this week, we're going to look, first of all, at a bunch of approaches that allow us to explore much better than we do now what those processes actually are, okay? So let's focus on that. And so one of the problems we've encountered there is, and one of the problems I have with process structuralism itself, the tradition that I come from, is that it's very rigid. And the same applies to René Thom's catastrophe theory. It's sort of a top-down, rigid mathematical framework that's put onto the reality of biology. And it's very difficult to come up with a general theory that applies to anything specific. So top-down theories in this field, it's too messy, it's too diverse. There's always new phenomena. It just won't work, okay? Also, this is very beautifully described here in this short comment called the Evo comment by Denis Duboul, this problem, okay? It seems very difficult. This is an interesting quote. I'm going to go into a bit of detail here because it's interesting to take it apart, okay? So Denis is a traditional reductionist molecular biologist, and he's writing here about the future of Evo, about which he's very pessimistic. And he's writing, we face a modern version of the classical dichotomy between variation, the how question, and selection, the why question. And we may wonder how long this productive relationship will last. It's a really interesting quote. So classical dichotomy only goes back to Meyer in 1961, but never mind. Maybe Meyer is a classic. I think so by now. That's, of course, the proximate ultimate distinction that doesn't really apply, but it pains Denis Duboul as a sort of a closet positivist, that wherever there's a friction between perspectives, that's a problem for someone who wants to unify. And so that's one of the problems that Evo has. There is no unification. While it may consolidate itself and lead to an integrated theory of evolution, this desire to have a unification again, that includes the emerging mechanistic side, it could well split again into divergent trajectories, and that would be the end of Evo, basically. Or not exactly, but like a comet that returns closer to a planet every 100 years or so to fill itself with concepts and energy before leaving again for yet another journey. So he says Evo and Devo are going to split and only much later come back together. This theoretical antagonism between development and evolution might nevertheless become obsolete once the mechanisms of development are fully understood. You need to have some sort of theory of development that's very broad and all-encompassing, and of course molecular in his sense. And once the computation of various ontogenetic roadmaps will discriminate the possible from the impossible, thus telling us which form could evolve out of a given species. So the task of having an idea about organic form is going to be very difficult if we need a general theory like that. And he writes, in such a scenario, the next rendezvous with the comet will turn evolution into a predictive science. That would be great, but this may indeed take another century. This is a basic misunderstanding of what systems biology should do, this sort of top-down, big theory, unification. But in the meantime, and we actually wrote a reply to this comment. I wrote it with Manfred Laubichler and Werner Kallebaut at the time, which was entitled The Comet Cometh. And there, and I've shown you this slide already, we write, we propose something resembling a series of targeted expeditions into the vast unknown territory of configuration space. So this configuration space, remember, is constantly changing in evolution. So there's no point in coming up with a big general theory about it. It's either too general or too specific, as I said before. Although we cannot gain a complete overview of the landscape yet, surely we could gain a lot of exciting new insights by just having a look around. So science has always been done like this, This seems a sensible approach in light of the fact that nobody has ever explored this territory before. We call this the natural history of configuration space. This natural history, that's the way you do biology. No big theories. Bottom up, you go explore. In analogy to the real-world expeditions that have led humanity to discover new countries, continents, and even new worlds beyond our own planet. Such expeditions always preceded the systematic mapping of the newfound frontier, not only in natural history proper, but also in many other branches of biology and science. So for now, let's just go and look around, okay? So the idea is to sample genotype-phenotype maps in specific systems that currently exist to arrive at an idea about the map of the possible that exists right now. Not a theory, not a full understanding like Denis Duboul writes. You don't need this. You need to go and have a look first and see, is there anything to be seen? And so there's a lot of room for this sort of dynamic mechanistic explanation that I was advertising, because at the moment we are excessively still focused in Evo in developmental biology in general about mechanistic decomposition, genetics, and maybe some causal modeling also in ecology or in evolution. But we should focus more on the recomposition, sort of dynamical systems modeling aspect, to unravel the complex causal flow that underlies these genotype-phenotype maps. So recomposition is crucial because there are non-local causes in biological systems, and also, because you want to contextualize different causes. Like I argued when we talked about causation in complex systems. Dynamic mechanistic explanation is useful to characterize morphogenetic fields, processes, and you can use a biology in a box approach. It's totally cool to do that, okay? So this may be inadequate to understand life itself, a whole organism, its interactions with the environment and how that evolves, okay? These are very complicated questions, but there's an awful lot of configuration space ready to be explored in exactly this way. We haven't done much of this yet. So what I want to do today, as the first lecture in this module, I want to show you how you can actually use modeling for this recomposition and how we can explore it further over the next few years. We're going to move on afterwards to see how this sort of helps us with a bunch of principles to understand the evolution of processes better in more detail and in a more flexible way than process structuralism originally intended to. So it's basically, it's somewhere in between developmental systems theory and process structuralism, I think. And also, ultimately, I want to talk about the evolution, the homology of processes this week. So that's the program for this week. But today, we're going to talk about 3 approaches by which you can have a model that is appropriate for a dynamic mechanistic explanation. We haven't actually talked about how you get those models in the first place. I just showed you a few examples. And so the first sort of good, it's a very good approach, is the I don't care approach. It was pioneered by Stuart Kauffman, and he didn't call it I don't care, he called it the ensemble approach, where you take network models that represent a certain system and you just simulate millions, gazillions of them. And you see what are the sort of the general properties of certain classes of networks with a certain regulatory structure, variational properties. What are, evolvability, robustness. We're going to come back to that. Or you could go to the other extreme and measure everything. So you make a biochemical, a biophysical model, and you measure the parameters of the model very carefully. And then you build the model bottom up. Okay, so this is the other extreme. So in one case, you don't care about the details at all. And in the other, you care about every detail a lot. Okay, both of these have their pluses and minuses, but there is a sort of a compromise in between, which is you can measure what is easy to measure and then estimate the parameters that are really important in your model, such as the strength of regulatory interactions. And that is the reverse engineering approach. That as opposed to forward modeling, you're not experimentally measuring all the parameters in your model. Let me give you 3 quick examples of that, and then we'll see what they're good for or whatnot. And we're going to move back to the fly and the segmentation gene system, and especially the maternal coordinate genes and the gap genes for these examples. Okay, so you have a sort of a gradient-based patterning system that looks a bit like a French flag. You have a gradient, and there's some thresholds in the concentration of the gradient, and they induce different sort of territories of downstream gene expression. And you want to understand how this process works. So the 3 ways to go about it is, first, you can just make a generic sort of model that simulates a morphogen gradient. And this is work by James Sharp and James Cotterell, beautiful work published in 2010, where they took a sort of a model of a tissue, a bunch of cells arranged in a line, and they have networks in them, 3 gene networks. The 3 genes are free to interact with each other in all kinds of different ways. Come back to that in a second, and they react and interpret concentration of a morphogen gradient. So you're sort of exploring which kinds of different networks, how do they interpret the morphogen gradient in principle, all kinds of networks. And so what they did is they were screening in silico, in the computer, for networks that could produce a stripe. One of the 3 genes needed to produce this sort of stripy expression pattern in response to the graded distribution of the upstream signal. Okay, so what they did is they varied the interactions among the three genes in all kinds of possible ways. So basically, look at these two interactions, alpha and beta. They can be positive and negative. I've introduced this sort of formalism before. So a negative interaction is a repression and inhibition, and a positive interaction is an activation. And so when the interaction is 0, you're on one of these lines. When it's, beta is positive, you're over here. When it's negative, you're over here. And so you can explore the entire parameter or configuration space of this model, doing massive amounts of simulations. And so if you subdivide this parameter space, you can find different topologies. And so you get a discrete space, which they call topology space, in which different network diagrams live here that are qualitatively different in at least one interaction from each other. And so you can look at all of those possible combinations and say, okay, which ones are the ones that can make a stripe in response to a gradient? I would call this structure space, not topology space, but never mind. So basically, that's what they did. And they sort of built up this beautiful diagram where you see every dot here is a specific 3-gene circuit. And the complexity of the circuits goes from 9 interactions, fully connected, to these 4 interactions down here. So as you go down the diagram, the dots represent networks with less and less interactions. And then they mapped out those, the color here shows you how many of those networks. Remember, the parameters can have specific values within one of those topologies. And so they mapped out how many of them were actually making a stripe. The darker, the better, okay? So that's sort of a probability of hitting a circuit within a certain area of parameter space with a certain topology of the network that makes a stripe. And they find only 6 topologies here at the bottom. If you prune it down as much as you can, you have these 6 kind of stalactites that have pretty dark color till they're down here at 4, maybe. This one is a bit of an exception, but here this is very dark, but a bit more complicated. And so they identified these 6 possible mechanisms that can make a stripe, and they all differ in their dynamics of making a stripe. Here you can see a sort of a space-time diagram of their dynamics, and you can actually automatically classify them because the dynamics of forming the stripe is very different in all of them. But what's important here is that only 3 of those here, they named them incoherent type 1, feed forward, mutual inhibition, morphogen gradient. Only 3 of them have been found in reality. 2 of them in Drosophila. So here's the mutual inhibition, classic. You have an activation of 2 target genes and they mutually inhibit each other. We've been through that. Remember the giant and the croup green, and they were also green and blue like here. Okay, so we've seen that before. This exists in the real world. But interestingly enough, they find 3 mechanisms of making a stripe that haven't been found yet and that are not very intuitive. If you look at those circuits, you don't immediately understand how they work. So what this does is it gives you prediction that you may find such networks, and it gives you help in understanding why they make a stripe. Because if you would find a network like that as a geneticist, you wouldn't intuitively understand what those interactions can do. But if you do now, you can go back to this paper and say, hey, this is a stripe making circuit. I mean, you have to do some testing, of course, if it really does it. Because of all the things we've been through. Not all of those networks will make a stripe, but a lot of them will. Okay, so this is interesting. This is an atlas for stripes, a map of the possible for stripe making. And so this sort of ensemble approach allows you to screen for, all the different mechanisms that could produce a certain pattern, given certain assumptions you have in the model, of course, in the setup of your screen. This allows you to map the space of all possible mechanisms for a certain pattern in an unbiased and exhaustive way. It was published in 2010, exactly the same year Denis Duboul was saying, oh, we're not going to have this map of the possible forever. On the downside, well, this is typical, right? If an expert says you can do something, it's usually a fair bet to believe them. If an expert says you can't do something, Dolly the sheep, cloning, a year before, lots of predictions, it wasn't possible. Don't necessarily believe them. Okay, so negative predictions, ooh, big grain of salt. On the downside, the solutions obtained are not too specific in terms of explaining your favorite experimental system. So if you're interested in some sort of specific bug in an organ and the bug and how it grows, it won't help you at all. So if you're that sort of type, it's better to go and measure stuff. And this has also been done in the Drosophila system. Let's look at the simplest example, and that is, how is this bicoid gradient actually formed? Remember, this is a syncytial embryo. The head is here, the tail is here, and bicoid protein is diffusing through the embryo. So you can measure the concentration of bicoid at different times in development, indicated by different colors, and you will see that the actual concentration of the gradient varies here. Don't look at the box here. This is a log-log rescaling, but just look at the sort of measurements here. And it changes over time, and it's a beautiful characterization. And you can then look at single positions in space, and then you see these are the mitosis that happens. So the nuclear concentration, of course, it breaks down and then it comes up again. Very complex dynamics. So you can characterize the spatiotemporal dynamics of this gradient in minute detail. Absolutely spectacular. And this being, these measurements show you that it's pretty close to a perfect exponential gradient. So you can model the system with the diffusion equation. Here, these are partial diffusion equations. It's a bit more complicated partial differential equations. It's basically a system that shows you the concentration of the morphogen C in space and time over time. Okay, so you have a diffusion term. Okay, so that's a change in bicoid concentration at position X over time T. This is a diffusion term, and shows you how diffusion changes the concentration across space. The bigger the difference between two nuclei, the bigger the diffusion rate. Bicoid protein decay. Okay, it's a very unstable protein. I don't know if you remember that. And then there's a source term. There's an RNA here, remember, that produces the protein. And so this is a simple enough equation that you can solve it analytically. Okay, and it has this sort of solution. Don't worry about the math too much. The only thing you need to understand here is that the shape of this gradient only depends on the ratio between diffusion and decay rates. That's it. And of course, the position of the source. But basically, if you know these three parameters, you can predict the shape of the gradient. And so here's a simulated, is a solution to this, and then juxtaposed with some of the measurements. So it's very precise. Okay, so this is fantastic. Three parameters we need to know about the diffusion rate, which was measured with sort of photo bleaching experiment and stuff like that, very precisely. It's a bit more complicated. There's a diffusion coefficient for inside and outside the nuclei, because the protein is supposed to get trapped when the transcription factor is in the nucleus. Half-lives were measured equally. Sort of, you see, this is a very short-lived protein, 10 to 35 minutes only. And then also the source term, beautiful work here by Thomas Gregor's group in Princeton and in Paris by now, measured the RNA distribution, just like we had the protein distribution before. So they measured everything here, beautiful, in huge detail, and they could fit the equation perfectly. And so at about this time, around 2010, we thought we understand the formation of this gradient perfectly. It's a diffusion that's sort of inhibited by the nuclei that trap the bicoid protein when it goes inside, because it's a transcription factor. But so if you eliminate the motive that makes the protein go into the nucleus, what should happen is that the diffusion is faster outside the nucleus. The gradient should change because it depends on the diffusion rate. And people have done these experiments and nothing changes. So this result sort of stands there, and people are debating still today whether this is a valid result or not and what's going on. But we're still, even in this super simple system, even if you don't understand the mathematical details, I went over everything quite quickly. What you need to understand here is this is the simplest possible biological pattern forming system you can imagine. No growth, no cell membranes, just a transcription factor that diffuses from a relatively simple source in one dimension. Wow, doesn't get any simpler than that. 10 years of efforts to measure it, and we still have problems understanding even a simple system like this. So this is where Denis de Boule's grim pessimism came from. I guess if you're a molecular biologist and you think you need to understand everything down to the molecular level, it's hopeless. So general theories are hopeless, like tome, process structuralism, and measuring every molecule, describing every enhancer. It's just one damn thing after another, and you will never, ever get there. And also a lot of the details that you study will be very context dependent and not generalizable at all. So you won't understand anything. Okay, so there's a problem. So forward modeling, this sort of precise measurement of all the parameters, enables us to study the molecular mechanisms of pattern formation using very rigorous quantitative biophysical methods. That's fantastic, but it's very labor intensive and requires a range of sophisticated experimental approaches for measuring parameters. We have more and more of that, so that's less and less of a problem. But in the end, it's difficult to apply to transcriptional and other complex gene regulatory networks. Okay, so we don't know the molecular mechanisms of eukaryotic transcription. We don't know how it precisely works. Okay, and gene-gene interactions are hard to measure using existing experimental approaches. So it hasn't been scaled up very much from this sort of gradient formation problem to more complex problems. It doesn't scale. Okay, so the compromise, of course, between the sort of everything, let's just simulate all these different networks and looking at a very precise specific instantiation is the reverse engineering approach, where you measure something that you can easily measure, like the expression of a gene, where it is. And then, and I've explained this to you, I hope you remember that you can just have a network model where you have this row of cells, the cells divide, you have gene-gene interactions, and you can fit the model to the data. And what you get out of this from fitting to these easily measurable gene expression profiles is sort of a matrix that gives you the structure of the network and a phase portrait of the dynamical model. So if you cleverly formulate this model, if you get the right kind of data set, this technique is extremely powerful to give you a prediction of the interactions, a network reconstruction that, unlike static network reconstructions, gives you the entire causal flow in the system. So we've been through this in several past lectures, but you can see this beautiful network of gap genes, and I can not only tell you what each of these interactions is, a repression or an activation, but I can tell you what it does at each time point and point in space. Okay, so this is a classical sort of dynamic mechanistic explanation, I would say. And so I also showed you work before that revealed this interesting sort of dichotomy between the anterior and the posterior of the embryo and Drosophila, and that these very similar looking patterns are built by very different mechanisms, sort of switches that go to different attractors in the anterior, and this sort of oscillator, this really broken clock in the posterior. I refer you to the past lectures for the details. What I want to illustrate here by repeating this is that this is very powerful, but it's not been very widely applied. It's still very difficult to do. I mean, I worked for about 20 years on gap genes. We've had to pioneer a lot of the quantification methods, the fitting methods, et cetera, but by now it should get a little easier to do this. And also, I'm going to talk a little bit in the next few lectures about an effort that we did to move out of the model system of Drosophila and move into non-model organisms and apply this to a comparative approach. It's very powerful, okay? And so what this showed is that, of course, the situation is much more complicated than this French flag model. So we now have a causal mechanistic insight into the genotype-phenotype map in a very limited example. So the phenotype here is just an expression pattern. Ultimately, what we want is a phenotype that's an actual form. Okay, so we're still very far away from that. And so I would say, okay, there's a lot to be done here. Okay, let's go and look around. What can we do with these three approaches? The ensemble, the forward modeling, and the reverse engineering. And we start to get a feeling which one of those is best applied. So reverse engineering provides a middle way between the ensemble and the forward modeling approaches. It is closer to the data than the ensemble approach. It provides specific insights into your favorite developmental system, unlike the ensemble approach. And it can be applied more widely than forward modeling because you're cheating. You're estimating the parameters that are difficult or impossible to measure, and you fit the model to data that is easy to measure, like expression data. less labor intensive, and allows you to estimate parameter values that you cannot measure even in principle, like these overall interactions between genes. But unfortunately, it requires some non-trivial math and data quantification. It's becoming more common to do this. And it has not yet been applied to many developmental processes. And what I find really frustrating is in developmental and biology and Evo, is that not a lot of, I mean, I hoped over the 20 years that it would catch on a little bit more, but not a lot of people are even trying to do this. So this is what we should be doing. If we want dynamic mechanistic explanations, we need more models like that, okay? We need to use the modeling techniques that we have, and we should start to come up with more and more examples, because by sampling genotype-phenotype maps in this way, so we sort of fill in the causal mechanistic details of the genotype-phenotype map with these intermediate levels of organization, these dynamical regimes, different mechanisms that are causal, can be at different levels, don't have to be at the molecular level. So let's start filling out this picture, sample more and more of these. And then out of that, surely will come some sort of general picture. What is common, what is not? I mean, if you think about segmentation in particular, there are not so many ways in which you can subdivide an embryo in 1 or 2 dimensions, okay? So there must be regularities that come out of the natural constraints. And this sort of general idea that Tom gave us, that in general, the way to, the number of ways in which you can break symmetry in general is kind of limited. It's not endless forms most beautiful in that sense. There is a certain order and structure to it. There must be. So let's go and discover that, not like process structuralism from the top down, but by actually studying empirically different morphogenetic fields, sampling them and comparing them, find out what they have in common and where they differ. So next time, we want to dive into this structure of the genotype-phenotype map, and I want to talk about a bunch of structural properties of the dynamic networks, causal mechanisms that are active here and that are influencing the dynamics of evolution. So I hope you tune in again next time, and thanks for listening, as always.